Hello, I'm Anishka Eliasheva from Department of Biology and Ecology at University of Ostrava.
Hello, I'm Bojana from the same department, and you're about to see our poster Plastic Cleistogamy in response to competition and drought stress.
Cleistogamy is the ability that some plants have to produce closed, obligately selfing, and open, potentially outcrossed flowers. Cleistogamy is often plastic, and the proportion of closed flowers can vary in response to environmental cues. In Lamium amplexicoli, the production of open flowers can vary between 50% in warm, long days and up to 100% in cold, short days. It is assumed that closed flowers are favored in resource-poor and stressful environments, which limit the plant development, whereas showy, open flowers are produced when the plant has sufficient resources and low stress exposition. Another non-exclusive explanation of this variation is that cleistogamy is an adaptation to pollinators' abundance. When environmental cues indicate high pollinators' activity, open flowers would be favored, whereas closed flowers, which pollinate autonomously, would be favored when environmental cues indicate pollinator-scarce environment. 10 years ago, I studied 4 natural populations of Lamium amplexicoli in France. 2 came from central or northern France, where the climate is temperate wet, and 2 from southern France, where the climate is Mediterranean and dry. In each region, one population came from what I called a favorable habitat that bears virtually no other vegetation and its soil is regularly plowed, so there is no competition with other species. Plants in these habitats grow big, with many axes and flowers. The other habitat I named unfavorable. It was densely colonized by other vegetation with putatively high levels of interspecific competition. Lamium amplexicoli is only rarely found in such habitats, and when present, the plants grow very small, usually with 1 to 3 flowering axes. In both climates, the large plants from favorable habitats produced about 80% of cleistogamous flowers. Surprisingly, small plants from unfavorable habitats with high interspecific competition had lower percentage of closed flowers regardless of the climatic differences. This interesting observation rose several questions about the variation of cleistogamy observed in natural populations. Is this variation an adaptation to environmental stress caused by interspecific or climatic variations? Is it an adaptation to pollinators' abundance among different habitats? And finally, is this variation due to phenotypic plasticity or genetic differentiation among populations? 10 years after the observations of natural French populations, we tested this hypothesis in a competition experiment that used seeds from 7 natural populations spanning an even wider geographic distribution and climatic gradient, from the continental Czech through Mediterranean Macedonian to desert Israeli climate. Whenever possible, we tried harvesting populations from the 2 habitat types. The seeds harvested from natural populations were grown in a common garden where we combined 2 unfavorable environments, interspecific competition and drought. The competition was simulated by growing the plants in a common garden in locally undisturbed vegetation, and plants in the drought treatment were rarely, if at all, watered. Both of these treatments are expected to reduce available resources and decrease stress, because the species is a very poor competitor that thrives in temperate climates. Unsurprisingly, competition and drought both significantly decreased plant vegetative growth, but their effects were not purely additive. If we look at cleistogamy, we see that the production of open flowers is low when the plants are watered, regardless of the occurrence of competition. However, when plants are suffering drought, They increase the production of closed, inconspicuous flowers. Most remarkable is the fact that competition always decreases plant size, but it only affects cleistogamy when Lamium amplexicoli is also exposed to drought stress. Investing in costly, open flowers in a competitive environment might be a way for the species to ensure that it produces outcrossed offspring, which is genetically more diverse and suffers less in breeding depression. Thus, the offspring can better withstand competition. However, this is only possible if the plant is not limited in water resources. Let's look at the pollination success now, which was measured as seed production. In closed flowers, pollination success decreased in response to competition, but also when the plants were watered. This is a very surprising result, given that seed production in closed flowers is usually independent of environmental conditions, as it has been shown in many cleistogamous species, including in a couple other studies of Lamium amplexicoli. Seed production of open flowers, however, did not respond to competition, but we could not estimate it in the drought treatment. This could be because there was no variation in the pollination environment. All plants were grown side by side within 10 meters of several beehives. Note, however, that despite the abundance of pollinators, closed flowers are always more efficiently pollinated than open flowers, which could go in favor of the reproductive assurance hypothesis. Finally, we observe differences among populations in the mean and plasticity of their traits. For instance, the percentage of closed flowers is significantly lower in the Israeli population compared to all European populations, and it does not seem to vary in response to competition. Among the European populations, we could not discern any patterns related to the geographic region or the habitat type. However, there was significant variation among populations for mean percentage of cleistogamous flowers and for their plasticity. Notably, the production of closed flowers varied from significant increase in the competitive environment to non-significant differences in populations with either low or high kleistogamy. From all this, it shows that variation in kleistogamy is due to both phenotypic plasticity and genetic differentiation, although it is not possible to devise a clear-cut effect of the climate or the habitat type. For all other traits, we observe similar results in all treatments. What is the takeaway message of all this? Let's review the questions. Is kleistogamy an adaptation to environmental stress? Yes, it is. We detect similar patterns in our common garden experiment to those observed in natural populations. When the plants are not limited by water availability, Lamium amplexicoli favors the production of open flowers in competitive environments. Is variation in kleistogamy an adaptation to pollinator abundance? Could be. Closed flowers produce more seeds than open flowers, but we did not test for the effect of pollinator abundance in our experiment, and we don't test explicitly about the quality difference of open and closed flowers. And lastly, is variation in kleistogamy due to plasticity or genetic differentiation? It is both. There are possible effects of climate to population differentiation, given the differences between Israeli and European populations, but we couldn't detect any explicit effect of the habitat type. I would like to thank you all for your attention. If you have any follow-up questions after the conference, don't hesitate to contact me by email. And I would also like to thank the talented Natalia Aguilera Lopez for preparing the illustrations for this presentation.